Okay, any questions? I don't think we need this. Let me state that I did a bunch of grading over the weekend and today. So there were a number of earlier labs, like maybe lab 2, 3, and 4 that got graded. And obviously the last lecture worksheet has been graded. And there's one other thing. I don't remember if it was a quiz or what, but oops, I'm losing my headphone here. Anyways, the point is take a look at that.
So what if our
I got the last lab graded and you can update your answers to that by Saturday, this Saturday. What's your question?
so what if our previous labs, like the ones you're saying 2, 3, and 4, have only 0.01 left? Does that mean we didn't get full credit or we didn't get partial credit?
If it's got either a 5 or a 0.01, it means I've only graded it one time.
Okay.
So hopefully that's not going to be lab 2, 3. And I don't remember if I finished grading lab 4, but I at least got started on it. All right. Can you see my screen?
Yes.
Okay, thank you. So today we're going to be working on lab 9, identification of gram-negative organisms. And you're also supposed to be working on your unknown work. Some of you haven't gotten started on your unknown work, and I'm not sure you're going to have time now to get it done because the unknown work must be due by March 5th. So if you haven't gotten started in your unknown work, you need to get started right away because you may not have enough time to get it started with. There was one student who asked me some questions and I tried to help her get started. All right, let's take a look at lab 9. Lab 9, the identification of gram-negative bacilli. You have some reading to do, not very much this time, before you do lab 9. There will be some video clips in the lab. Your learning objectives. Once you do this material, you should be able to interpret the results for the following biochemical tests. The IMVIC test, and IMVIC stands for the indole, methyl-red, Voges Percure, and the citrate test. So IMVIC are 4 tests that are very commonly used with the Enterobacteriaceae, that family of bacteria. You should be able to understand the results for carbohydrate fermentation and the motility test. Now, along the way, we're going to discuss hydrogen sulfide production, and there's 3 different ways you can assay for hydrogen sulfide production, but you should have an understanding of the results because all of them look dark if it's positive for hydrogen sulfide production. Be able to explain and interpret a TSI test, that's triple sugar iron. Be able to explain and interpret the Sims test. And both of these tests look at more than one, well, test or one thing at a time. Be able to explain and interpret the growth on a McConkey's plate and hectone enteric agar plate. These two plate, McConkey's or MAC, and HE or hectone enteric, assay the same things, but they have different colors, meaning they both are selective for gram negatives, and they both can are differential on looking at lactose fermentation. Be able to use the biochemical analysis to solve various case studies involving gram-negative bacterial infections. So you'll have more case studies today. All right, members of the family Bacteriaceae are very common small gram-negative bacteria. They're non-spore forming bacilli, but they're small bacilli. They're all facultative anaerobes. They all ferment the sugar glucose, and they're all oxidase negative organisms. There are 53 genera of the family Enterobacteriaceae, but 24 of the genera are known to cause human infections. And here are the common ones that you'll see causing a bacterial infection. Most of these genera are normal microbiota of the intestinal tract, aka coliforms. So we call them coliforms, and all it just means is if it's a coliform, it's something that can grow in the colon or the intestines. They are all opportunistic pathogens in that they can cause other infections, or they can grow outside of the intestines, such as the urinary tract, wound infections, pneumonia, septicemia. Some common genera that cause opportunistic infections include Escherichia, such as E. coli, Enterobacter, Klebsiella, Citrobacter, and Serratia. So these are common in the intestinal tract of people or humans, and they can cause opportunistic infections when they're not growing in the intestines. Several genera are associated with gastroenteritis. These include organisms in the genera Salmonella, Shigella, and then specific variants of E. coli, such as Serovar 0157H7, a human pathogen. It is E. coli, but it's a variant that is a human pathogen. Salmonella and Shigella, if you don't know, are not normally growing in humans. They're not a part of our normal microbiota, but they can grow in your intestines, and then they'll cause disease. Pseudomonas aeruginosa is similar to the family Enterobacteriaceae in that it is a gram-negative bacilli. However, this is a different family, the Pseudomonas family, and all the members of this family do not ferment the sugar glucose. They're all oxidase positive bacteria. Pseudomonas aeruginosa causes opportunistic diseases, so it's an opportunistic pathogen. It is particularly a problem for immunosuppressed individuals, burn patients, and trauma patients, meaning Pseudomonas aeruginosa very rarely infects a healthy human. It's not good at getting established on a healthy human, but an immunosuppressed patient, a burn patient, or a trauma patient, Pseudomonas aeruginosa can colonize those regions and then grow, causing an infection. It's also common for causing health-acquired infections, or HAI, and we're going to discuss that in the lecture, but in the textbook, it's called a nosocomial infection. So nosocomial infection and healthcare acquired infections are the same thing. It's important for microbiologists to be able to identify and perform antibiotic testing on gram-negative bacilli, especially once they've been cultivated for clinical specimens. In this lab, we're going to discuss the common biochemical tests that are used to identify gram-negative bacilli. And we're going to look at the two families, Enterobacteriaceae, and then the family Pseudomonaceae. The first test we're going to talk about is the indole test. Some organisms can produce the enzyme tryptophanase that converts the amino acid tryptophan into the products indole and pyruvic acid. After overnight incubation, Kovacs reagent is added to the tube, which will react to indole to produce a red color at the surface. E. coli tests positive for indole production. So the amino acid tryptophan, it's converted by the enzyme tryptophanase into indole and pyruvic acid. And then that's not looking very red. We add Kovacs reagent and it's positive if you see red at the top. Methyl red and Voges Proskure tests are two tests that are normally started together in the same tube. And then the tube is divided into two parts, one part to do the methyl red test and the other part to do the Voges Proskure test. Any questions about any of that? The methyl red test and the Voges Proskure test, or the VP test, are used to detect fermentation of glycolysis. If acid products are made, we then use the methyl red test to look for acidic products. But if neutral products are made by the fermentation of pyruvate, then we use the Voges Proskure test, or the VP test, and look for the neutral products of the VP test. So here we have glucose, which goes through glycolysis to make pyruvate. It can make acid products, and we can use the MR test to look at those acid products. And it can make neutral products, and we use the VP test to detect those neutral products. All right, the MRVP test is incubated for 2 days to 5 days. Let me see if I can remember this right. The VP test is only grown for 2 days. The methyl red test is grown for 5 days. And if you read about it, it's 2 days and or 5 days. If it's negative, the MR test has to be repeated and you run it for 5 days. After incubation, the methyl red test, you added methyl red to the tube, and the methyl red is a pH indicator that turns red under acidic conditions. And if the tube remains red, then the acids from the mixed acid fermentation occurred, and it'll keep the methyl red red, and then the tube is positive. Any questions about the methyl red test? If not, we'll move on to the VP test. The VP test looks at the neutral products of glycolysis. And if they're made, it will turn red when it's positive. Negative is not red. You have to add Barrett's A solution to the tube and Barrett's B solution to the tube. Mix the tube to add oxygen to the tube. And then let it sit for 60 minutes. And if acetone is made from glycolysis, it's a neutral product of glycolysis, the tube will turn red. If it doesn't turn, and then it's VP positive. If it doesn't turn red, it's, if it doesn't turn red, it's VP negative. And that's the VP test. All right, citrate utilization test is looking at the utilization of the carbohydrate citrate, where citrate is the only energy source in the tube. If the bacteria can utilize citrate, like in the Krebs cycle, the bacteria will grow on the tube and turn the tube blue. If the bacteria do not utilize the citrate, then the bacteria will typically not grow and the color of the tube will remain green. All right. Any questions about that? E. coli and Shigella can metabolize citrate and they will turn the tube blue. The next test we need to talk about, which is not part of the IMVIC. IMVIC stands for indole, methyl red, V for Voges Proskura, the VP test, and then the, I just talked about it, the IMVIC, the C for the citrate test. Carbohydrate metabolism is looking at the fermentation of a carbohydrate in the tube. If the carbohydrate is fermented, it will produce acids, and then the color will change from red or pinkish to yellow for an acid product. You can also have an inverted Durham tube in the carbohydrate metabolism test, and you can look at the production of gas from the fermentation of the sugar. And if you have the production of gas, you get a bubble in the inverted Durham tube. If you have an acid, meaning the sugar is fermented, but there is no gas produced, there will be no bubble in the inverted Durham tube, but the tube will turn yellow from the acid. Any question about that? All right, for the carbohydrate metabolism test, you have one sugar in there, and that sugar, if it's fermented, will be yellow. And we call that A for acid. And if it's acid and gas, we call that AG. In the case of a negative fermentation, we then mark that negative. There can be different carbohydrates that you can test, such as glucose, sucrose, lactose, mannitol, inulin. There will only be one sugar in the tube. And we usually call that like the PR lactose test, the PR glucose test. But here we're just calling it the carbohydrate metabolism test. And there will be one sugar in there to test for the fermentation of that sugar. And you could test for all of them. You just have to run different tubes that have different sugars in them. All right, any question about the carbohydrate metabolism test? If not, let's look at the motility test. The motility medium is a semi-solid media, meaning it does have agar in there to solidify it, but the agar is very low, so it's semi-solid. And if the agar is low enough that if the bacteria have a flagella, they can swim out of where you stabbed it. And that happened in this tube where if you notice, let me blow that up a little bit. Of course, it moved it. I don't seem to be able to blow that. Not sure. Oh, I know. See why I can't blow it. See if I can blow it now. Nope, it's not blowing. Anyways, here we stabbed it here, and then the bacteria swam because it had a flagella, and then the entire media is cloudy, as opposed to this region of the media here, and this region here, and this region here. It's not cloudy, it's clear. And in this case, the bacteria did not swim, and it's non-modal, and so it's only growing in the stab, which we can easily see here. So this one non-modal, this one is modal. Now, if you have a bacteria that's only slightly modal, you may not see it cloudy everywhere in the tube. What you may see is that the bacteria is growing around the stab, and you'll see it in a feathered-like position. I'm trying to draw it, where it's protruding from the stab mark. And that's just because it's modal, but it's not very modal. All right, that's the motility test.
So would you describe that as like a line through it?
It's not a
Clear?
line through it. I tried to draw it here. I was trying to get it bigger. Let me see if I can just copy this. It's not letting me do that. Oh, there it is. So we have the tube like this. And it has a stab where you put the bacteria. And the bacteria are growing more than where you stabbed it. It'll be growing. It'll be kind of hard to do. That brush will be better. It'll be growing like this.
Oh, okay.
Growing away from the stab. And they call that a feather-like position. It may not be perfect in alignment.
So it's just clear with like feathered.
Yeah, it's growing out from the stab is the point.
Okay.
And there's the growth there. And then this region here will be clear because the bacteria haven't swam there yet.
Okay.
Okay. All right, let me go back to where I was. All right, the hydrogen sulfide production test. In the presence of hydrogen sulfide, which is made by a bacteria, the reagents will turn black and you'll see a dark color. And you'll notice it's not black at the very top. And that's because the hydrogen sulfide is only made under anaerobic conditions. So it'll be made usually in the lower portion of the tube and not in the upper portion of the tube. This one, most of the tube is black, but sometimes you'll only see it black in the very bottom of the tube because that was the only region of the tube that was anaerobic. Hydrogen sulfide, for at least most species, is not made in the presence of oxygen. You can test the production of hydrogen sulfide in different media. You can test it in triple sugar iron, also called TSI. You can test it in sulfide indole motility, which that's so long, almost everybody calls it a Sims test. You can test it in the HE auger plates or the hectone enteric auger plates. And in all the cases, if hydrogen sulfide is produced, it will turn black. What happens is the bacteria produce hydrogen sulfate, and then the hydrogen sulfide reacts to the reagents in the tube. I believe it's iron, and then that forms a black precipitate. Citrobacter, Salmonella, and Proteus produce hydrogen sulfide, and they will turn the tube black. An organism that doesn't produce hydrogen sulfide will not turn the tube black. In this case, the color is unchanged. And in this case, it's also a yellowish color. But for the TSI tube, if it's hydrogen sulfide production, you'll see black in the bottom of the tube. Usually it's not black at the top of the tube. And the non-colored one would be the non-hydrogen sulfide production will be red in the TSI. Here it's here. So non-colored and no reaction is reddish. And if the hydrogen sulfide production, it's black. We'll talk about the yellow when I get there. Here's black in the Sims tube and not black. And no hydrogen sulfide is being produced. With hectone enteric auger, if you have black colonies, you have hydrogen sulfide being produced, and non-black colonies, no hydrogen sulfide is being produced. All right, let's talk about the triple sugar iron test, also called the TSI. It is an auger slant with the butt in the auger. There's 0.1% glucose, but 1% lactose and 1% sucrose. So if fermentation occurs, it will be yellow, but you don't know which sugar has been fermented. It could be glucose, could be lactose, it could be sucrose. And we'll talk a little bit more about that. There's also a pH indicator, it's phenol red, and that's what determines whether the color is reddish or yellowish. It turns yellow into acid production. And if the sugar is fermented, it will turn yellow. It also has ferrous sulfate and sodium thiosulfate, which will turn brown if hydrogen sulfide is produced. You don't need to know that. What you do to inoculate them for the TSI tube is you get a needle and you get the bacteria on the tip of your needle, and then you inoculate the bacteria. You should go down a little bit further than this is showing. You should go down about 2/3 of the way from the bottom of the slant to the bottom of the tube. And that looks like it's closer to about halfway. So you should go down about 2/3 of the way. Why you're doing the stab is you want to look at the bacterial growth growing anaerobically. And that's why you want to go down 2/3 of the way. At least 2/3. You usually don't want to go all the way down, because if you touch the needle or the needle onto the glass, you'll damage the needle, you'll bend it. And then you take the stab out, the needle out, and then you pick up fresh bacteria. You just roll it in the colony, and then you inoculate the surface of the slant in an S-like, snake-like fashion. And this is where you're looking at the bacteria growing aerobically. And then you grow the tube and look at the color of the tube and what's going on in the tube. And I'll provide this picture and this to show you. If there's no change, the media stays reddish or pinkish. The hydrogen sulfide production will be black at the bottom of the tube. Let's talk about this one first. I'll get back to that one. And if the agar turns yellow, you have fermentation of the sugar. Any question about that? Which sugar is fermented is kind of hard to say. That's tube 4. You have either lactose or sucrose fermented, or both. You don't know because it's all in one tube. And you have glucose that's fermented. Glucose is the base sugar. And if lactose or sucrose are fermented, or both of them, then you always have glucose fermented. In this case, we have gas that's produced because there's a bubble down here. The agar is broken, so gas is produced. No hydrogen sulfide is produced because the tube's not black. And we know that lactose or sucrose are produced because the tube is yellow, both on the bottom and on the slant. If the tube is only yellow on the bottom, but not on the slant, we know that only glucose is fermented, because if lactose was fermented, it's present in 1%, and it would be yellow both on the slant and the bottom. If glucose is only fermented, it'll be yellow on the bottom because the bacteria grow slower. The glucose is metabolized slower in the butt. But on the slant, the bacteria can grow rapidly, and the glucose is initially fermented. So initially, if you're looking at this tube after a few hours, this would be yellow. But as the glucose is all used up, the bacteria will start fermenting protein and then releasing ammonia from the amine group of the protein. And that ammonia is basic, and that will remove the yellow color and then turn it reddish. And that's simply because glucose is only at 0.1%. And so it gets used up on the slant, and then you'll lose the yellow color and it will revert back to red or reddish. So tube 3, only glucose is fermented. In tube 4, glucose is fermented because it's yellow, but another sugar is also fermented because it's yellow on the slant. And that sugar will be either lactose or sucrose or both. We don't know. Any question about TSI? All right, so TSI is complicated because you're running more than one test. We're looking at glucose fermented. We're looking at glucose and one other sugar fermented. We're looking at hydrogen sulfide being produced, and we're looking at gas production. And all of these are going on at the same time. In the SIMS test, the sulfide indole motility test, you're stab inoculating the tube with a needle. And then you're looking at hydrogen sulfide being produced, in which case the lower portion of the tube will turn black because hydrogen sulfide is produced only in anaerobic conditions. You then add, you look at the stab to see if the bacteria are only growing in the stab, like tube 2, or if the bacteria are growing away from the stab, tube 4. And this one, the bacteria are motile, and that's why it's black everywhere. Any question about that? So this is motile, and you only can tell from the black. This one is not motile. That one is hard to see, but it's not motile. And tube 5 is not motile, but tube 4 is motile. Only have a growth is away from the stab. And then you add Kovacs reagent to the top of the tube. And if it turns red, let me see if I remember now. Then you had is that VP acetone? Sorry. Oh, it's indole. Yeah, you add Kovacs reagent to look for the production of indole. And if it turns red, it's positive for the production of indole. Indole is made from the breakdown of tryptophan, the amino acid tryptophan. If it's negative, the color is unchanged, yellowish. And here, because black is everywhere, you can't tell if it's yellowish or if it's red. Actually, it's red on the top. Sorry, I don't know what I'm thinking. It's red on the top, so we do have indole being produced. No indole, no indole. And then indole is positive, indole positive, indole positive, indole positive. All right, any questions about the SIMS test? Like I said, it's a complex test. You're looking at the production of indole, you're looking at the motility of the bacteria, and you're looking at the hydrogen sulfide being produced. McConkey's and HE test are tests looking at multiple things. They're both looking at the growth of gram negatives. If the bacteria can grow on McConkey's or HE, it's a gram negative. If it doesn't grow, it's a gram positive. We then look at the fermentation of sugar. On McConkey's, if lactose is fermented, you'll get a pink color. If there's no fermentation of lactose, it's yellowish or whitish color. On HE, which we don't have a negative, but it's no orangish color or salmon. If it ferments the sugar lactose, and if it doesn't ferment the lactose, it's non-salmon colored. You can look at the, you can look at the hydrogen sulfide being produced on HE agar. And if hydrogen sulfide is produced, you get black colonies. And this is a non-lactose fermenter. And if there's no hydrogen sulfide, then the colonies are not black, they'll be whitish or off-white. All right, any questions about MacConkey's and HE? So in this lab, you're not going to be physically doing any of the biochemical tests. We're going to do it all online. You are responsible for knowing how to perform and interpret the tests. So we have a video on the carbohydrate fermentation test, another video on the SIMS test, and a video on MR and VP2 tests, a video on triple sugar iron, TSI, and then a video on MacConkey's agar. You also have to view some of the website Microbuzz, and that's to complete the step 1 of worksheet lab 9. You have a table here, which I don't even have it here. Let me look at worksheet 9. So you have to fill out the table, the positive result and the negative result. Oh, come on. I don't have the arrow over here, so let me. I don't know why. There it is. So you look up oxidase test, indole test, and tell me what the positive result is for oxidase is filled in for you. The cells or the spot turns blue. The negative result is not there. I'll give it to you. The cells do not, or the spot does not turn blue. It stays its original color. So look up the table to get the results, fill it in, and you can view it or using the website Microbuzz. You do, once you get to Microbuzz, you have to click on the differential test off on the left, and then click on the specific test of interest. So differential test, and then we're looking at, I don't know, what's one we look at? Probably citrate, because we're talking about that in this lab. And then you can read about citrate test here, and then look at a positive result and a negative result. Any questions about any of that? Once you're done filling out the worksheet to lab 9, which has the table, then answer the questions in part 2, and then complete the case studies in part 3. This table is, you use this table to identify the bacteria in the case studies. Do not use this table to identify your unknown bacteria in the unknown project. Only use this table for the case studies in lab 9. And then you have the case studies right here. You can fill in the table here for what the different tests were. And then for case study unknown 1, tell me what the species is that caused the infection for case study 1, 2, 3, 4, and 5. Okay, you need to tell me what the species is. You can't tell me what the family is, because all of the infections of this lab probably are going to be happening by Enterobacteriaceae or Pseudomonaceae. Any questions about any of that? All right, go ahead and get started on the lab. I will be here until 8 o'clock to help you if you have any questions. If I'm not in the camera, it means I stepped away from my desk for a few minutes. Like I think I want to get something to eat, as well as maybe get something to drink. All right, start on the lab. I'll be here until 8 o'clock to answer any questions.